And I'll do this. Hopefully that bar isn't in the way. Okay, so good night. Again, welcome to the Unit 1 Celebration Review. For the people on the Monday group, you already did a review with Dr. Cotter. If you're in the Wednesday group, we'll go over a review on Wednesday during lecture, and then I believe she has something scheduled after lecture, or if you finish early, she might schedule it in between our lecture time. First things I want to highlight, just generic information. So the exam is worth 80 points and you have 90 minutes to complete it. The exam will be a combination of multiple choice questions and short answer questions. Also have some drop downs. And you can start taking the test Thursday morning at 7 in the morning, and you'll have up until Saturday night at 11:30 p.m. So around the same time your case study would be due. You do need Respondus lockdown browser for this. So if you have not done it, I don't believe Dr. Cotter's doing a 2-point assignment, but I believe she had a Respondus assignment at the beginning of the semester. But if you have never used Respondus before, just let me know. I'll send out an announcement for how to set it up and how to test it. But you do need Respondus lockdown browser for this. If you can't use it because your computer can't handle it, or maybe you're not in a good testing environment at home, a little bit too loud, etc. If you can afford, they're welcome to come to the library and you can just take the test there. Or we have quiet rooms you could rent out and you can take your test in that quiet room and then finished up. That way you have reliable internet and it's also a quiet place to take your exam. If you are going to do it on an iPad, you can do it on an iPad. I made a YouTube video for that, for how to access it and log in. I should have that announcement out already, but if not, I'll resend it out if you want to take this exam on an iPad. And biggest thing is if you do have any issues when you're taking the test, please let Dr. Cotter and I know as soon as you can. Probably Dr. Cotter, because she can actually fix most of your problems. I can do some of the technical stuff, but anything that requires like redo of the exams or reopening a test, that's going to be all Dr. Cotter. So just reach out if you have any issues. And this kind of goes back to don't wait till the last minute to take the test. If it's Saturday at 10 o'clock, I don't know if you're going to get that response back before the exam closes. So the sooner you figure out your problems, the better. And another thing that we want to highlight, this is recorded. And so what Respondus will do is it'll let us know if there's excessive noise, if there's excessive movement, if your eyes aren't looking at the screen, or if they're gazed away, if your head's turning. It sends little flags every time you do it. And then once you're done with the exam, we get a summary of all the students and it tells us red, yellow, green. So green, the respondent said, okay, you're good. You probably didn't do anything weird. Red, it flags you as potentially cheating or doing something like that. And then yellow, the respondent software can't determine what you did, but there was something that really caught its attention. And then we have to go back and look at the recordings and figure out what happened. One of them, someone was taking their exam during dinner and we could hear their parents in the background talking. And so respondents flagged that because there was constant noise while they were taking the test. Just don't do that to yourself. Don't do that to us. Just find a quiet place to take it. Take your exam. I know it says you have 90 minutes. I've seen on average, most people get done around 40 minutes or so. So less than an hour, get the exam taken care of in a quiet place, and then go ahead and go about your day. Okay, so the way this is going to work is I'm going to go through key highlights in the case study or things that I have seen other students mess up before on the exams. That way you can learn from their mistakes and ideally you don't make the same mistake. One of the first main mistakes that people make is they forget Koch's postulates. We're already in week 5, maybe week 6. And by this point, we've all kind of forgotten about the Koch's postulates. So again, just to remember what Koch's postulates are, remember to observe the patient for signs of illness and isolate the microbe from the infected host. If you get asked a question about step 1 in the process, make sure you also list out what the signs are and what the symptoms are. Don't just say, oh, I would check the patient for signs of illness and isolate the microbe. Also say the signs of illness were they had a puncture, they had a fever, took a temperature of 102 degrees, whatever it might be. And then the symptoms, say the patient was complaining about a sore throat, they were complaining about a headache, whatever it might be. Make sure that you specify what the signs and symptoms are that we are giving you. That way you can maximize the credit for that question. Part 2, so culture and identify the microbe in the lab using different tests. This was one that was missed a lot in the case study. So page 17 of the yellow handout gives you a nice little list of all these tests. The biggest thing you have to do with this one is select 2 tests. So microscopic, macroscopic, for example, and give a very brief description of what that test is testing for. So if you say microscopic examination, you're trying to identify cell shape, cell size, etc. If you say macroscopic, you're determining colony morphology, colony color, that kind of thing. Just those very brief little descriptions to add to your answer, again, to maximize your points. If you put something like biochemical and you don't know what biochemical tests you're going to test for or what chemicals you're going to test for, just don't put it down. Put one of the other 2 that we've worked with. We work primarily with microscopic and macroscopic. You should have multiple examples for that one. I would just go with the one that, it is easy, but you'll get the points for that versus missing points trying to answer one of the other ones. Again, don't forget to mention that you're going to infect a new host with the same microbe you isolated, and you're trying to observe the same signs of illness. Again, we're mostly nowadays we'll do testing like on cultures, like we'll actually grow some stuff. We're not going to test it on live animals or live people, but make sure you put that step there. And then again, for step number 4, you're repeating the same test from step 2. This is also missed on the case study where people gave us 2 tests on question 2 and then 2 separate tests on question 4. They have to be the same tests. So as long as you use the same 2 tests for question 2 and question 4, you should be able to get full credit on this one. Okay, so cell parts and cell part functions. The biggest thing with the cell parts and cell part functions in regards to the exam is the exam's going to work a lot like your case studies. So I believe it's going to be 3 or 4 different cases, and it's going to give you a couple of different things. So for example, I might say, we isolated your microbe and we noticed that we had a viral spike was seen under a microscope. Okay, so we know that viral spikes are part of viruses. So you shouldn't be putting, oh, that could potentially be a bacteria or a fungus or a protozoan. You should be able to say, oh, based on a viral spike, that should have one of the 2 different types of viruses that we talked about, naked, enveloped. We also talked about the bacteriophage. But just know that viral spikes, if given that information, you should be eliminating bacterial cells and fungal cells. Same thing if we give you things like cell wall, right? Is it peptidoglycan cell wall? Is it a chitin cell wall? The cell parts that we're testing for, it's not going to be like a definition. Here's the cell part, give me the definition. It's more of, we're going to give you a list of cell parts, and you have to tell us, oh, these parts are for a bacteria, these parts are for a fungal cell. Or even more specifically, these parts are for a yeast fungal cell versus these parts are for the other type of fungus cell, which I cannot remember right now. But you get the gist of it. Is the cell parts, we're not testing for what the cell parts do. We're really testing for what the cell parts are part of in terms of the grand microbe. But if you do want to look at the cell part functions, here's a good general description of the cell parts. This is all BIO 181, 182 stuff. Again, what we're looking more for is if we give you cell parts, can you identify what type of microbe those go with? Bacterial shapes, you might be given some images that show you a bacteria under a microscope, and that's going to help you determine your case study. Some of the questions that could get asked are the flagella type, the location. If I give you a picture of this top one, you got to make sure it's monotrichous, or if we give you different shapes, you should be able to identify between Staphylococcus, Streptococcus, Streptobacillus, Bacillus shapes, right? The main shapes that we've talked about in lecture and in lab, you should be able to identify that if we give you a picture. Again, this is a big one. So like I said, the exam is 80 points. You're going to have, I believe, anywhere between 16 and 20 questions, but some of those questions are going to be compare and contrast or drop down. So we're going to give you Let me see if I can have a good example of that. Okay, so I'll use step 2 of the study guide. I don't know if you can see it. Hopefully you can. Yeah, okay. So page 2 of the study guide, you have this awesome chart comparing eukaryotes, fungi, and viruses. Ideally, anything on that we could ask you for in one question, saying, hey, given these cell parts, we want you to put this part with this cell, or this part with this prokaryote, or this part with eukaryotes, or even something like what's a good one here? Cytoplasm, right? If we give you cytoplasm, that could be eukaryotes and prokaryotes. So that would also be a drop-down option. So your drop-down options could be none of the above, virus, prokaryote, eukaryote, eukaryote and prokaryote, eukaryote and virus, virus and prokaryote. You get the gist of it. The biggest idea is that we're going to give you drop-downs and you have to match the cell part to the specific microbe. Again, going back to what I mentioned earlier. I know it doesn't seem that difficult, but you gotta remember we made what, 1, 2, 3, 4, 5. There's 5 different charts just in the case study alone for cell parts and microbe parts and what type of microbe they go to. So any of those could be asked as a question, and that one question could be worth multiple choice or multiple different points. And I do have a question in the chat. Is spelling a big part? The only time that we really care about spelling is going to be the microbe. So like on your case studies, you're going to have the case, work your way through. We're going to give you something very simple, like you see cells, or you see that it's non-living, something very specific to tell you it's okay, it's probably a virus, a fungus, or a bacteria. And then we're going to give you extra information to then break it down. So if it's a bacteria, you should already be anticipating it's going to give you extra information to determine whether it's gram-negative or gram-positive. If it's fungus, we're going to tell you it's going to be yeast or it's going to be mold. Or if it's virus, we're going to give you extra information for you to determine, okay, I need to go the naked virus route or the complex virus or the enveloped virus route. But the only time spelling is going to actually count is going to be when you answer the question. It's going to say, based on all this information, what do you believe your microbe is? And that's where you get the genus, species spelling. For the viruses or the microbes, when you go to that question, it's going to give you a list of potential suspects like you get already with the case studies. So you don't have to memorize how to spell these microbes. We're going to give you a list of microbes already in the question. It's up to you to determine which microbe it could be. The big ones for the microbes, the stuff that we've already dealt with, Staphylococcus aureus, E. coli, influenza, HIV, the yeast and the molds. If you go to page 7 of your study guide, we gave you a nice little list. Let's see if I can see it, of different fungus, protozoans, parasites, and gave you some examples to write down there. Any of those examples could be on the test. So if you really want to prepare for those, I would go back and just go through either your unit one, your book, go through your notes, or even just Google some of these and see what some of the standard ways to diagnose these are. That way you can answer them. Worst case, if you don't know the microbe name specifically, you're still going to do well on the exam. It's not worth as many points as the other questions are leading you to that point of answering the question. But that was a good question there. Okay, viral replication. So I think this is probably one of my last slides. This one is one that's going to get a lot of people. It's already gotten people on this last study or case study. 2 things with viral replication. We've already given you 2 different viruses that we're going to work with, HIV and influenza. So you need to be able to identify based on if we give you hemagglutinin or GP120, you have to recognize, okay, GP120, HIV, I got to go the HIV route. Or influenza, hemagglutinin, I need to go that route. You have your 6 steps. So you're going to have a question on the 6 steps, I guarantee it. And you're going to be asked for specifics. When we look for specifics, when I say step 1 attachment, if your microbe is influenza, any type of description with attachment should start with hemagglutinin viral spike is used in attachment. For entry, neuraminidase enzyme is used for attachment, or sorry, for entry. Replication, another one that was kind of commonly missed was people would say viral DNA or RNA. Okay, well, we know that's DNA or RNA, but is influenza an RNA virus or a DNA virus? So you gotta be specific, saying it's an RNA virus and it replicates using RNA polymerase. HIV, what makes it very special is that it uses reverse transcriptase. So HIV unloads its RNA, it then has reverse transcriptase to reverse write it into DNA, and then the DNA gets then transcribed into RNA and then to proteins. It makes it very unique about HIV, but just know for replication, HIV, reverse transcriptase, influenza, RNA polymerase. When you get into assembly, you start saying, oh, I'm going to assemble all the parts of the virus and make new virions or virions. Make sure you mention what the parts are. So you're going to say, oh, I'm making brand new hemagglutinin and neuraminidase viral spikes. I'm going to add reverse transcriptase, and I'm going to add RNA from the HIV virus to put these new virions together, right? It's all about being specific with these questions, not just writing your 6 steps and not just saying generic terms like, oh yeah, it's used for attachment. Okay, what's used for attachment? Again, be specific. That's all I really have. It's not a lot to go through. Biggest thing for this one is anything on the Unit 1 study guide is fair game for the test. So if you have any blanks on the Unit 1 study guide, what I would start with is making sure that you could answer all of the questions in here. And then if all you do is study the study guide, you'll be fine on the test. People do tend to overthink it a little bit for this class. Dr. Cotter doesn't like to overthink. She gives you everything that could be on the exam on the Unit 1 study guide. Make sure that's filled, review that, and then you should be all set. Other than that, make sure you have good Wi-Fi, have a quiet place to work, have a reliable computer. That's basically it. Very quick, very painless. Again, I just wanted to go over the biggest things I see that are missed commonly over the last 4 years. And it's always cell parts. It's always the viral replication, and then Koch's postulates for some reason. They're very simple, but we just kind of forget about them. And I'll check the chat. And then someone said, for future reference, will I be doing reviews like this before exams? Yes, so I will be doing an exam review for Unit 1, Unit 2, Unit 3, and Unit 4 traditionally gets combined with the final exam. So I'll do a final exam / Unit 4 review. But yes, I will have a review. I think Mondays before the exam will be the day I choose. Rose, most of the questions, it'll be a combination. So you're gonna have a combination of multiple choice and drop down. And by drop down, I mean like you click on a little box and it has 5 options, and you're gonna click on one of the options. And then the other part of it could be a separate option. And then you are gonna have some typed ones. I guarantee Koch's postulates question is going to be a typed one. You're going to put, okay, step 1 is this. This is what I would do. These are the signs. These are the symptoms. Step 2, these are the 2 tests that I'm going to do. Step 3, et cetera, et cetera. And then when you're answering the question of what is the genus, species of your microbe, you're going to also have to type that out. But besides that, multiple choice, drop down, and a few short answer. But the short answers are things like I mentioned. Viral replication is probably going to be a short answer. Writing out the steps, writing out the specific parts of the virus, and that kind of thing. Hopefully that answers your question. And Sarah, on the steps, it's going to only be on influenza or HIV. Yes, we're not going to throw so the question, it goes to viral replication. We're not going to throw a random virus that we just found off the internet. It's going to be either influenza or HIV. So study both. And I'm guaranteeing you'll get one of those 2 on the exam. Okay, yep, no problem. Do you have any other questions from anybody in the call? Okay, if I don't have any questions, I'm gonna get kicked off here by Zoom in any minute. So thank you very much for attending. Like I said, I'm going to upload this recording, and you can go back and look at this again if you want to. Thank you very much. Enjoy the rest of your night. Again, reach out if you have any questions. Wednesday group, I'll see you on Wednesday. Monday group, reach out to me like you have been, and I'll get back to you as soon as I can. All right, thank you.